Good morning. I hope everybody can hear me. Welcome to the Cytek commercial tutorial. My name is Maria Jaimes. I'm the Director of Technical Scientific Support at Cytek. At past CYTO meetings, I have had the pleasure to talk to you about our technology and explain to you the advantages of moving flow cytometry towards the full spectrum approach. Today, I'm really excited to have two of our users present and show you how they have been using and applying our technology in their areas of research. So in the first part of our tutorial today, we will have Dr. Aymeric Sylvain present on his data on research done on COVID studies, looking at the immune response in patients. Dr. Sylvain works at the Institut Gustave Roussy in Paris. And then in the second part of our tutorial, we will have Dr. Eric Davis from MD Anderson walk us through a different application and how they're measuring FRET using a spectral flow cytometry. So without further ado, it's a great pleasure to introduce Dr. Sylvain. And Aymeric, the floor is yours.
Thanks, Maria. I would like to thank Cytek and CYTO 2020 to give me the opportunity to present you the work that we did in Gustave Roussy during the COVID-19 period, which happened between March and May in France. And during this period, we basically used a lot our Aurora machine in order to build up the paper entitled Elevated Calprotectin and Abnormal Blood Myeloid Cell Subset Discriminate Severe from Mild COVID-19 patient. And in order to explore what was happening between infected patient and non-infected patient, but with flu symptoms, we managed to perform single-cell RNA-seq on 6 patients. And we also performed spectral cytometry on 115 patients. We performed CYTO only on 12 patients due to the time-consuming experiment. And finally, we also performed cytokine and chemokine quantification on 84 patients. So just to give you a quick overview, what happened is that we were receiving blood samples from these patients. And it was in an emergency state, so I didn't have the luxury to have time to build up my panel. And I quickly built up a panel with 25 markers and that I found in the fridge. So basically, it was already amazing to have 25 markers. And this thanks to the Aurora. It was quite easy to find 25 fluorophores that were not overlapping. And I started to pass the samples on the Aurora. And then after the acquisition, what I did, I checked the compensation. They were really good already with beads. And I modified them much less than what I used to do on a regular fax. But in order to have a good representation of my cell subset, I used a non-supervised method to cluster and to reduce the dimension. So we call it UMAP. And from this UMAP view, we could clearly identify neutrophils, CD4 positive T cells, CD8 positive T cells, B cells, monocyte, and NK cells. And then what was amazing is when we started to split between control patients, infected patients with mild disease, and infected patients with severe disease, we could clearly see that some cell population were highly moving, meaning that some markers were changing. So basically, if you look at the neutrophil between the control, the mild patients and the severe patient, we could clearly see that the neutrophil profile was changing. As well, for the monocyte, we could clearly see that between a control and a mild, there was few differences, but between the control mild and severe, the monocytes were changing. So then we zoomed inside the monocyte and came back to a regular representation of the facts plot. And here it's all the patients concatenated together. So you get an overview of the mean of what is happening in all the patients. And basically what we observe in the patient when we were looking at the 3 different subsets of monocytes, we could observe that in severe patients, non-classical monocytes were decreased compared to controlled and mild patients. And when we look inside the classical monocyte, we could observe that these classical monocytes were losing their HLA-DR in severe patients. So here you have the representation in bar plot, and it's significant. The loss of non-classical monocyte was significant. The loss of HLA-DR was significant. And then when we look at the neutrophil, it was obvious that the percentage of neutrophil was increased, but we also checked in absolute number, and it was also increased in patients with severe COVID-19. And then when we look at the phenotype of this neutrophil, what was striking is that in severe patients, neutrophil were losing CD10 and CD101, suggesting that they were more immature. So this was just to have a very quick overview. It was the first sample that we analyzed to see which population was changing. Then we decided to do single-cell RNA-seq, and for that, since we saw that the neutrophils were highly modified in severe COVID-19 patients, we decided to do 10x single-cell RNA-seq, but we did a very quick red blood cell lysis on fresh blood samples. So that's why we had so few patients, because we had to work fresh in order to keep the neutrophils to have them in this single-cell RNA sequencing. This single-cell RNA sequencing revealed that in a mild patient at day 0, we observe a very strong interferon alpha signature with interferon-induced genes that was lost 10 days after the day that this person presented itself at the hospital with symptoms. And then in the severe patient, meaning that they were in ICU, we observe a completely different signature. We observe that neutrophils were overexpressing CXCR4, MME, and most of all, one of the highly overexpressed genes was S100A8 and S100A9. And it was really highly expressed. It was the top DG in neutrophil in severe patients. So in order to validate these results, we quantified the cytokines inside the plasma of these patients. And when we compare controlled plasma to a mild patient, we observe that the only cytokine chemokine that was overexpressed in mild was interferon alpha. And when we compared control versus severe patient, we observed a bunch of cytokines that were highly overexpressed, and the top cytokines was Calprotectin, which is the dimerization of S100A8 and S100A9, the top DG that we had by single cell in the neutrophil in severe patients. Here you have the bar plot of the cytokines. We could see that CXCL8 was decreased in mild patient compared to control and severe. We observed that IL-6 was overexpressed in severe, but it was only 4-fold more. and interferon alpha was highly increased in mild patient compared to control and severe. And the calprotectin was strikingly increased in severe, and the fold change is between 100 and 1,000 times more. So calprotectin compared to IL-6 seems to be much more increased in severe patients. And then we decided to validate some of the targets that we could by spectral cytometry. And here is the validation. So same thing again, we performed with a new panel, the analysis. We could observe again on a bigger cohort this time because we had in total 48 controls, 16 mild and 26 severe. We could observe also the loss of CD10 and CD101 in severe patients. And when we look at CXCR4 expression, we could observe that CXCR4 expression was increased only in severe patients, suggesting that immature neutrophils were released from the bone marrow in severe patients. And in order to confirm this data, we also performed some patients on CYTO, and it took us 2 days of acquisition, while the spectral cytometry with the Aurora took us 1 full day for all the patients. And we observed also with the CYTO that neutrophils were losing CD10 and CD101 in severe patients. Then we focused also on the monocyte by single cell. And here again, in mild patients, we could observe a very strong interferon alpha signature at the days that the person presented itself at the emergency with symptoms. And after 10 days, the person didn't have any more symptoms and was completely fine. And this signature was completely lost in the monocytes. In comparison, and one of the gene that was overexpressed and which is an interferon alpha inducible gene was Cytek-1. And you will see later, we tried to validate it with using the Aurora cytometer. And when we checked in severe patients, monocyte, we could observe that they were overexpressing THBD, thrombomodulin, and also CXCL8 and NF-kappa B. And we also tried to validate these targets. So here is a representation of the percentage of non-classical monocyte, like I showed you in the first part of this presentation. In severe patients, we could observe that severe patients had a loss of non-classical monocyte in their blood flow. So here you have a severe patient with their time in ICU and the percentage of non-classical. And we observe that most of them lost their non-classical and during the first week at the ICU. And then later on, some of them were recovering their non-classical monocytes. Some of them were undergoing bacterial infection, but it doesn't seem that it's linked to bacterial infection. It seems that it's also linked to the fact that some of these patients were going better and would get out of ICU a few days later. But here you have a representation. You can clearly see that non-classical monocyte decreased the first week of ICU. And then after a week spent in ICU, they come back. And here you have the representation. And when we look at the classical monocyte fraction, we observe that among the control, only 3 among the 48 controlled were expressing Cytek-1, CD169, while in the mild patient, 11 among 16 were overexpressing Cytek-1, suggesting that what we observed in the single cell RNA-seq, the overexpression of Cytek-1, we could validate it using the Aurora. And finally, in the severe, some of the patients were overexpressing it, but most of them were not. And then when we look at the thrombomodulin, which is also CD141, we observe that in severe patients, we clearly had an increase of expression of thrombomodulin, which is also, that plays also a role in thrombosis. And here you have the quantification. You can clearly see that thrombomodulin increase in severe patients, while Cytek is highly overexpressed mostly in mild patients. And finally, it's a very, of course, like there is much more results than the one that I present here. We did correlation. We show that Calprotectin was correlating with fibrinogen and didymers, which are thrombosis factor. And Calprotectin was also correlating with the amount of neutrophils that we could find in the blood. But most interestingly, we tried to see if by quantifying these different parameters, meaning Calprotectin, non-classical monocyte, and immature neutrophil, we could predict if the patient was going toward a severe COVID-19. And here you have a ROC analysis that unravels that Calprotectin is one of the best markers to differentiate a severe patient from a mild patient. And in the article, you will see we also have moderate patients. And Calprotectin in moderate is intermediate level between mild and severe. The non-classical monocytes also is a very good way to determine if a patient is moving toward a severe COVID-19. We had a lady who showed up at the hospital, and we ran the analysis, and when we realized that she had no more non-classical monocyte in her blood, we called the doctor saying like, this person doesn't have any more non-classical monocyte. And in fact, the lady came back the day after because she required oxygen. So it really seems that this kind of analysis using cytometry can help in order to the hospital to perform their triage and to know which patient has to be followed and which patient can be sent back home. And the CD16 low neutrophil seems to work too, but I would be less enthusiastic about it by flow because neutrophils can be tricky. And in our design, it is important to understand that all the samples were put in cytodelix, which is a solution that allows to froze the sample and maintain the neutrophils. So that's how we managed to analyze them by fax. And then, of course, in the discussion, we discussed the fact that Calprotectin is a very good target. It has been shown to induce myeloid emergency. And it seems that it can also induce an immunosuppressive response. So we propose to use an anti-calprotectin drug, which is called takinismod, that has been already tested in a clinical trial phase 2 of a metastatic prostate cancer. And here is basically the hypothesis of the result that we got. We splitted mild and severe. What we suggest, the data suggests that in mild patients, you have a very strong antiviral response, interferon alpha mediated, and that the immune response remain functional, meaning that you have classical monocytes that express their HLA-DR, you keep mature neutrophils, and you can still have non-classical monocyte in the blood. While in a severe COVID-19 patient, the soluble mediators that will be overexpressed are calprotectin, IL-6, and CXCL8, which are a symbol of very strong inflammation, and I think we can call it a chronic inflammation. And this chronic inflammation could induce an emergency myelopoiesis, and this will induce the appearance of classical monocytes that lost their HLA-DR, so that are more immunosuppressive, the appearance of immature neutrophils that secrete a huge amount of calprotectin, and the loss of non-classical monocytes. So this is our discovery about COVID-19. I would like to conclude that Gustave Roussy is a cancer institute. So we basically use the pipeline that I showed you in COVID-19, but it's a pipeline that we will use also in cancer in order to establish new targets and to find innovative treatment for our cancer patients. What the ability of Gustave Roussy to allow us to have access to numerous human tumor samples is a huge opportunity in our case, in order to better understand what are the immune response that are deleterious and what are the immune response that could help in order to eradicate tumors. And finally, here, we used the Aurora, and really I used to do cytometry, conventional cytometry. I used to do 16 colors, and it was already quite difficult to compensate the samples. And I would say that the Aurora is so much simpler. After, I'm sorry to say, there is no cytometry for dummies. You need to understand how a cytometer works. You will need to check a bit your compensation with the Aurora, but really, I was able in an emergency to build a 25 color panel easily. And to be honest, you can go until at least 35 color. And Cytek will help you, and Cytek helped me a lot, and in my learning and in my design of my panel, and even like to find solution. So I would say that spectral cytometry is the future of cytometry, and it's a very amazing tool. And as we complete it with some algorithm to reduce the dimension, but it's really great. And finally, this COVID-19 project has been feasible through a huge collaborative work between Paris hospitals and Gustave Roussy. We were 48 people who worked all together during 4 weeks, because we had only 4 weeks to generate the experiment, because we received the first sample the 6th of April, and the first draft of the paper was submitted on the 8th of May. And then the lockdown was not anymore there, so Gustave Roussy went back to cancer research. So it was a great opportunity and a very stimulating experience. So here you have all the people that I would like to thank. So like you can see, it's a really huge collaboration between Gustave Roussy, APHP, even Singapore, and even like Israel with Idoamit. And of course, we thank all the funding that we receive in order to make that possible. Thanks a lot. And if you have any question, I can answer if we have time.
Thank you, Aymeric. Yes, we do have a couple questions. The first one, you mentioned that the paper will be published soon. They were asking if you could share the reference.
So the paper will be online this week in Cell. It's not yet online, but they told us it will be in 2 or 3 days.
Thank you.
So as soon, the title of the paper is the title of the presentation.
Okay. Another question, can you comment on any steps you took for optimizing your panel?
So, for example, in my case, I did some few mistakes the first time that I used. since you have Alexa 488 and you have BB515 that you can use at the same time on the Aurora, do use markers that are really far from each other. For example, CD4 and CD8. If you use two fluorophores that are really close to each other, but the Aurora has the power to split them, it's important for you to use not markers that are co-expressed by the same population, but markers that are really far from each other and that will stay in two populations that are really far from each other. So very strong protein, but really like Cytek, if you have a panel and you want to optimize it, Cytek has amazing people that helps. You can send your panel that you try to build. They have also a beautiful app, a website, which is called Spectrum. You can go there, choose your fluorophore, then create your similarity matrix, and then you just have to look at numbers. If the numbers appear and they are bigger than 0.98, you cannot use these two markers together. If they are below it, but they are at 0.95, you have to be careful. You have to choose, for example, CD4 and CD8, or LY6C and CD3, two markers that will clearly stain two different populations. But really, like Spectrum is an amazing tool to build your panel. And after, depending on your marker, try to not use colors that are close to each other for population, for markers that are co-expressed, to make sure that you do not have an issue with compensation. And you can check your compensation, but like, I don't know, like if some of you use DAPI on a regular cytometer. For example, DAPI and Pacific Blue is a nightmare to compensate. And sometimes you reach 80%, 100% of compensation. And you wonder if it's true or if it's not true. With the Aurora, you will never reach this kind of value, even after when you look at your compensation. So it's you feel much more confident about your compensation, to be honest. Me, in my case, it was PBMC. I use beads. I think that where you have to be careful is when you use cells that come from a tissue that has been digested. Maybe it's better to do your monocolor on cells and not on beads for this kind of tissue, or at least once to make sure. But otherwise, for PBMC, the beads worked really fine. I just check after, modify the bead, but it was nothing drastic.
Thank you, Emeric. So I think in the interest of time, we'll move on. So Maria, back to you.
Thank you so much, Dr. Sylvain, for such a beautiful presentation, and looking forward to your publication. So before we move to our next speaker, I would like to take a few minutes to show you some tools and some recent developments that we are doing at Cytek in order to empower you with the use of the full spectrum cytometer. So as Emeric was talking about panel design, he was explaining, and as many of you who are listening to this tutorial, you're familiar with panel design, and you know that the panel design process takes certain concepts into consideration. Everybody knows that you need to match the fluorochrome brightness with the antigen density, and that we need to watch for spillover spread. And as Emeric explained very nicely, understanding antigen co-expression is key. If you want in-depth explanation of our panel design approach and how we recommend to optimize panels, I will highly recommend that you go to our website, and on the resource page, you will see a webinar that we did very recently, and where we explain step by step our approach at optimizing highly multiparametric panels. Now, I want to go back to the first point in this process of panel design, and is the fluorochrome selection. So in conventional cytometry, you are restricted by the optical configuration of the instrument. But as Emeric explained, not anymore with full spectrum cytometry. We are in such a flexible space now, and you have so many more considerations, and brightness and spread are one, but something that is going to be very key is understanding which fluorochromes work well in combination. And everything starts with this beautiful, beautiful image that is an information that we get in our instrument that is the full spectrum. Many of you, and we have showed these over the years, what the full spectrum is, but basically, this is the fingerprint of every fluorochrome, and those up and downs tells you what is the peak of emission, but also areas where you are going to expect spillover and where you need to pay attention to your panel design. So over the years, and because we are really strong at education and making sure that flow cytometries have as much information as possible to make sound decisions, we have developed multiple tools to allow you to better understand the fluorochromes and to assess fluorochrome compatibility. So I'm going to walk you very quickly to historically the resources that we have developed, the tools that we have developed. So first, we came with our beautiful fluorochrome selection guidelines. They're available through our website. Then, as Americ explained, we have a really, really nice spectrum viewer. And that spectrum viewer is also available through our website, and different to other spectrum viewers, these show you the full spectrum, and you can have a very good understanding of how the fluorochrome is behaving across the full spectrum after excitation with every single laser. And then, as Americ also alluded, we have now progressed, and beyond visual inspection of the spectrum, what we have are metrics, numbers, because I know it's easier to convey cutoff and numbers. And so we have developed two indices, the similarity and the complexity indices, that will allow you to have a very, very specific understanding and a metric to tell, are two fluorochromes the same? So that similarity index will be very high or very different. And how a combination of dyes work when put together. And that's what the complexity index does. Today in this commercial tutorial, I don't have the time to go in depth, but I will really, really encourage you at this virtual CYTO to please listen to the presentation of my colleague, Huiming Hu. You will see here at the bottom of the slide appear the name of her presentation and how to get to this presentation. Please listen to it. It's a beautiful presentation, and you will understand exactly why we feel so strong about the similarity and the complexity index. So we have spent tremendous amount of time characterizing dyes, understanding their spectrum, and understanding how they work together. So it was a logical step for our company to now start developing our own reagents, because now we understand so much about the fluorochromes and how they work together. So we're very excited to introduce a new family of reagents. They are going to be named C-fluors. And the nomenclature of the name is very simple. It's the name of the family of reagents, C-fluor, followed by the initials of the laser, that is the primary excitation. In this case, for example, C-fluor YG for yellow-green, and then the peak of emission 584. And we have, I'm showing you the name of 3 dyes that we have launched, one excited by the blue laser, one by the yellow-green, and one by the red. The one excited by the yellow-green laser has been now used by many of our collaborators who are working on COVID studies. It's part of a 35-color panel that we're using worldwide to look at immune response in COVID. We are also working in many other prototypes dyes that are currently under development in R&D, and our goal is going to be filling the full spectrum and making sound decisions on the best dyes that can work together. The assessment of the dyes and what direction we want to go with these dyes is based on, as classically done by any manufacturer, looking at brightness, stability of the dye, spillover, but of course, we're very interested at knowing and understanding the multicolor performance of the dyes and how they play together in combination. So in the next slide, what I'm going to present you is data from a 14 color panel, and where we were aiming at looking at different monocyte, B cells, and T cell subsets. I know it might be difficult to read, but when you look at flow data, and as I have explained over the years, the quality of the data, you can assess the quality of the data by looking at the clustering of the double negative populations. And in these few plots that I'm showing you here, I hope that you appreciate how tight those populations are and how good is the resolution. Why? Because we are choosing dyes that play very well together, where you see in this matrix of the similarity index that we do not go very high in that index, and we keep the complexity index as low as possible. What we would like to do in our next chapter with developing reagents is to work closely with you at developing and doing panel design, as we have done with, and we will be doing hopefully with Dr. Sylvain, and plugging in our new reagents to your new panels, and hopefully you will enjoy and you will get this high quality data. So this is for now. For more information on these dyes and questions that you have, please visit our virtual booth, and you can always contact us. And I explained to you how we have been using the full spectrum to guide our development and our search for new interesting dyes. And now Dr. Eric Davis will tell us and will show us another way of using the full spectrum, and now it's detecting FRET. So Dr. Davis, all yours.
Thank you. Good morning, everyone. I also want to thank Cytek and CYTO 2020 for giving me this opportunity to present our work. The two people who did most of the work are Jared Henderson, who's a PhD student with me now, and Andrei Havranek, who was a postdoc and is now back in Czech Republic. So I'm going to talk about something that's quite different, very technical, but is one in which spectral unmixing is equally valuable. And so we addressed the question of using FRET. And this is shown here. The basic principle of FRET is, in contrast to the usual situation where an isolated fluorophore is excited and then emits that energy in its characteristic wavelength spectrum, if there's an adjacent fluorophore with the right characteristics, some of the donor excitation is transferred to the acceptor, which emits in its different characteristic wavelength. And there's several factors that determine the efficiency of FRET, but key is the distance between the donor and the acceptor. And this is a very dynamic effect, and many assays have been designed in which the FRET depends on the distance between the donor and the acceptor. And if that in turn reflects some property of interest, then you can have an assay based on FRET. You can measure FRET in a variety of ways, but most commonly it's by changes in fluorescence emission. So on the left, if there's not very much FRET, when the donor is excited, most of the fluorescence emission comes only from the donor, but when they're close to each other, there's less emission from the donor and an increased and acquired emission from the acceptor. And this is usually measured, can be measured with only 3 different channels of fluorescence emission. And this is the so-called 3-cube method. So it's necessary to measure the emission from the donor after donor excitation, and then also the emission from the acceptor after donor excitation. And this is the so-called FRET channel or FRET effect. But it's also necessary to measure the emission from the acceptor when the acceptor alone is excited in order to be able to make corrections for the abundance of the acceptor. You can simply measure the ratio of the acceptor emission to the donor emission, the IDA over IDD. This is the so-called FRET ratio. It's very simple. It's often applied to flow cytometry data, but it's insensitive, and it's also very dependent on the instrument and the conditions that are used. Instead, if one calculates efficiency, then you have a measure that's instrument independent. So the 3-cube method was developed to measure FRET by microscopy, which has certain advantages, very efficient at capturing emitted fluorescence, and you can also deliberately photobleach the acceptor, which allows some back calculation of how much FRET there is. And of course, microscopy provides subcellular localization. But there's some disadvantages to microscopy that are potentially solvable by using flow cytometry to measure FRET. So microscopy is fairly slow and laborious, so you can only analyze a small number of cells, so there's less precision, small number of samples, so there's lower throughput. The cells need to be immobilized, so there's really nothing that can be done with them other than to measure their FRET. And to my knowledge, microscopy generally does not measure FRET as well as the abundance or presence of other fluors simultaneously, or calculate FRET or efficiency in real time. So for my presentation, you need to know 2 things key. One is that there are kinase activity reporters that use FRET. And in this case, it's for the kinase AKT. And so there's an artificial molecule that has to be expressed in cells of interest. And this is a single protein that has the donor fluor at one end and the acceptor at the other. And in between, there's a linker that has a target peptide for the kinase of interest, and then has a domain that can bind to that target peptide once phosphorylated, which will then generate a higher degree of FRET. And so the degree of FRET from this reporter is proportional to the degree to which the reporter is phosphorylated, which in turn is reflective of the activity of the kinase AKT. And this is very useful as compared to other ways of measuring kinase activity. And you can see that there's a modular design to this reporter. So if we change the target peptide to be specific for a different kinase, then we can have a reporter for another kinase. And so based on that reporter, we need to have some other cells that are engineered to express control. So the donor alone, the acceptor alone, and then 2 separate controls in which the donor and acceptor are either very close to one another or held far apart by a rigid spacer. And these are then the high FRET and the low FRET control. And then finally, as a physiological control, we have a version of the reporter in which the target peptide has been mutated so that it cannot be phosphorylated. And so this is a so-called dead reporter. So when you use these controls and reporter cells and collect data in the 3 different channels, it's possible to calculate what the FRET is, but that is done after the fact. And with the measurements of the 3 channels, IDD, IDA, and IAA, there's adjustment necessary by some constants that are calculated based on the use of the single color controls and the low FRET and high FRET controls. And with all this data, we wrote some R software, and then it will automate the calculation of the FRET efficiency. So the second thing that you need to know is what system did we predominantly use this reporter on, and that is on signaling by the B cell receptor. So the B cell receptor is a signaling complex that signals in 2 different ways. One is at baseline tonically in the absence of cognate ligand or antigen. It activates predominantly a single pathway shown here, leading to the activation of AKT, but also in response to antigen or mimicked by the use of a BCR antibody for cross-linking, it activates AKT more strongly, as well as some other pathways. So here's what the results look like when using a conventional flow cytometer, measuring the 3 channels of fluorescence and calculating the FRET efficiency. If what's shown here is on this axis, the x-axis is the intensity of the reporter itself, or the abundance of the reporter, which varies between cells because we're using random transfection. And then the y-axis is the calculated FRET efficiency. It's based on single cell calculations of E, but those are very noisy. But if we use a moving average and measure thousands of cells, which flow cytometry can do, then we get very precise results. You can see in green is the high FRET control, in orange is the low FRET control, and in between are values for the reporter. In gray is the dead reporter, in black is the live reporter, and this difference is the amount of FRET that's attributable to AKT activity, and then in red is a cell with the live reporter in which the B cell receptor has been knocked out. And so even though these differences are relatively small, they're very precise and they're reproducible on multiple instruments, multiple days. And this is how we were able to demonstrate that tonic signaling by the B cell receptor activates AKT in these cell lines. But we can also use this assay in a lot of other ways. And here I show that you can use small molecule inhibitors of kinases in this pathway from the B cell receptor, in this case as an inhibitor of SYK. Shown here is the endpoint results at different concentrations of the inhibitor. In blue is the dead reporter, is the good control, and then in red are the values of FRET with different doses of the inhibitor. So you have a classical sigmoidal dose response curve. If we do the same thing using multiple inhibitors of AKT itself, we again see the sigmoidal dose response curve, and we can test 5 different AKT inhibitors in 2 different cell lines, and we get very similar classical results. This would not be possible to do this basically any other way. We can also use the assay in kinetic studies. So shown here in black, again, we have the dead reporter. In gray, we have the live reporter as a control. And then red, we have cells with the live reporter that have been exposed to a stimulus. And so this is versus time, like a calcium flux assay. If we add an inhibitor of AKT, you can see that within minutes, the activity of AKT as reflected by this FRET reporter declines nearly to 0. If we add a stimulus, in this case, it's a BCR cross-linking antibody, you can see that within minutes, you get a very strong response and increase in AKT activity. We can also do a longer time course and using endpoint assays, cells exposed, incubated for different amounts of time. And you can see here with the live reporter in red that we get a peak in AKT activity after BCR cross-linking at about 30 minutes, and it lasts for around 2 hours. So all those data were acquired using a conventional cytometer and post-facto calculation. So when we obtained a Cytek Aurora, we first tested whether we could get similar results using just comparable channels from the multi-channel spectrum on the Aurora. So shown here are 3 spectra. This is the low FRET control, the high FRET control, and unstained cells for background. And we know which channels were IDD, IDA, and IAA based on the laser that excited and the wavelength of the excitation. But you can also see here that there's a bump if you compare the high FRET spectrum to the low FRET spectrum, and that is the effect of FRET, and that is the IDA channel. And so if we acquired data from all these different channels, but just picked out the ones for the 3 channels and did our usual calculations, then we could generate efficiency values. And as shown here, with a variety of different conditions and levels of FRET activity, the results with the Aurora using the 3-cube method are very similar between the Aurora and a conventional cytometers, again, validating that, in fact, efficiency is an instrument-independent property. With the Aurora, we could get similar dose response curves using the reporter engineered to report not on AKT, but on protein kinase A over a spectrum of doses of a very strong agonist. So it gives similar results, but in fact, better results because there's more precision at the single cell level. On the left is a display of the single cell efficiency values with the conventional cytometer, and on the right is with the Aurora. We have dead reporter, live reporter, low fret control, high fret control, and you can see that there's much less spread of values measured with the Aurora. So at this point in the presentation, I can say that using flow cytometry with a fret reporter to measure kinase activity is a very good assay. It's fast, precise, accurate, cheap, flexible. You can measure the activity of different kinases. It can be done with a conventional flow cytometer. It's easy to use once you know how everything's available. And the same principle of fret detection, in theory, could be used for things other than a reporter assay, like protein-protein interaction. But this assay does have limitations, some of which are inherent. So, for example, it does require the use of a reporter. So cells have to be engineered. It doesn't provide subcellular localization, and it's not suitable for fixed cells. But there's some other limitations that can be overcome or might be using the Aurora or spectral unmixing. So I've already shown that the Aurora can give better precision at the single cell level. But also, I'll show that with spectral unmixing, we can use this assay with other fluors, and most importantly, we can calculate or we can measure fret in real time as opposed to efficiency, which can only be calculated post facto. And this is a major advantage. So to show how we did this, measuring or assessing fret by spectral unmixing is very simple. These same spectra for the low fret control and the high fret control, we simply have to designate as fluors, or we might call them pseudo fluors, and tell the instrument to use its software to linearly unmix the fluorescence of the reporters as a combination of low fret and high fret. And so this is to remind me, this is the money slide. So when we do this, if we have the Cytek Aurora perform fret unmixing, it looks like this. If we can take those two parameters and show them on a contour plot, obviously on the low fret axis, we see cells of the low fret control. On the high fret axis, we see cells of the high fret control. And in between is where we find cells with the reporter, either the dead reporter, the live reporter, or the cells with the live reporter that have been stimulated with this strong agonist. The same data, if we were to display in a fret ratio in real time, doesn't give anywhere near the same degree of separation of the different cell types. And this is true for a conventional cytometer or even on the Aurora with using the designated channels as a fret ratio. Now that was using a sledgehammer, the forskolin agonist with the pKa reporter. If we use a somewhat more physiologic stimulus, which is an antibody to the B cell receptor and the AKT reporter, we still get very good separation. So this is all the same data, but presented in different ways. The top is the fret ratio from the Aurora of cells that were either stimulated with anti-BCR in red or not stimulated in blue. We get some degree of separation with the live reporter, no separation with the dead reporter. But if instead we look at the spectral unmixing, which is available in real time, you get excellent separation of these two populations based on fret. Now, this was not actually done by mixing the cells run simultaneously, but they were run in parallel. So we tested whether, in fact, we could mix cells that were either stimulated or unstimulated and together analyze them. And we did that in different ratios. And you can see that the fret was able to separate cells in a way that matched what the theoretical ratios were of the proportion of stimulated and unstimulated cells. But we wanted to go further and verify not just by relative proportion, but by actual direct evidence that the cells that had been stimulated through the B cell receptor were the ones with the high FRET. And to do that, we first established that you can use a different fluor, in this case, AF647, which is very different from the donor and the acceptor of the reporter, and conjugate it directly to the anti-BCR, and it will not interfere with the measurement of FRET. So the bottom is shown the FRET over time in a kinetic assay of the dead reporter. Nothing changes even when cells are labeled with the anti-BCR. And then on top is the live reporter. The same increase in FRET is seen whether the anti-BCR is unlabeled or labeled with the AF647. And that's even true with 3-cube E measurements. You can also take the spectral unmixing data, low FRET and high FRET, and similarly calculate a ratio so you can plot that versus time. So establishing then that we could use a labeled antibody to stimulate the B cell receptor and not interfere with FRET for the AKT reporter, we either did not stimulate or stimulated with the labeled antibody cells and mixed them together and then analyzed them simultaneously. And you can see that these two populations live in different areas in the spectrally unmixed FRET contour map. And then just to prove that this is something that can be seen in real time, I'll show you a movie in which we're going to take AKT reporter cells and stimulate them with the labeled anti-BCR. So there they go. They are now coated in anti-BCR. The dead reporter cells FRET stays in the same area, but the live reporter cells move out of that area. It doesn't move quite as far as what I've shown you previously because that was a 30-minute endpoint assay in culture. But in real time, you can see that you can unmix, you can see cells with different FRET levels by spectral unmixing. So then it's an important question. I mean, Alexa Fluor 647 is so different from the donor and the acceptor, it's not really perhaps a fair choice. Could we use other fluors that overlap more with the emission from the acceptor, which is where the FRET channel lives? And here's PE and then dialite 488. So we took cells with the reporter and we incubated them in a kinetic assay, or just hit them in the kinetic assay with the antibody, either unlabeled or with these 3 different fluors. And you can see versus time, all the cells get very strongly labeled. If then post facto we calculate the FRET efficiency, you can see that with the dialite 488, there's an artifactual increase in FRET that's really just due to the fact that that fluor bleeds into the IDA channel. But with spectral unmixing, it doesn't matter. The instrument's able to calculate the FRET from the reporter independent of the fluor that's on the antibody. We did a more complicated experiment but more realistic, in which we took our B cell lines that have the AKT reporter with these 2 fluors and labeled them with antibodies that have these markers on them, all of which, except for CD3, are expected to stain the cells. So cells were stimulated in culture with the anti-BCR, then put on ice, which we found stabilizes the reporter signal, and then we stained them with these other antibodies and analyzed. And so we had cells that all the cells had been stimulated, but only half had been labeled with the various antibodies. And we took them through this assay and then plotted those that were stained with the antibody versus those that were unstained. And they have absolutely the same unmixed values in this FRET assay. So the assay, spectral unmixed FRET, can tolerate the presence of multiple other fluors.
Eric, we have about 1 minute left.
Okay.
Thank you.
And you could possibly use this, you can imagine you could use this in a genetically engineered mouse that has the reporter. Then I'll zoom through that. You could also use the reporter in a genetic screen, a genetic knockout screen, to identify genes that are important to the response to B-cell receptors. So if we have a unrelated antibody, CXCR4, we see that all the cells respond to the anti-BCR. If we knock out the BCR, which we can only do in a fraction of cells, we see that there are some cells that are unresponsive. And then if we knock out AKT, we also have an unresponsive fraction of cells. So in summary, measuring FRET by spectral flow cytometry is very simple. You only need low FRET and high FRET controls. You can use it with other fluors, and it provides FRET discrimination in real time, which is critical and novel. You could use this not only for reporter assays, but antibody-based studies and to measure protein-protein interaction. I think you could use it with in vivo reporters and in a genetic screen, which would require physical sorting based on FRET. That is the end of my presentation. Thank you.
Dr. Davis, thank you so much for such a beautiful presentation, and thank you everybody for joining us today in this commercial tutorial. Dr. Davis, if you could go to the last slide, please. Everybody, if you have questions, as we were not able to take them now. Next slide, Dr. Davis.
I was signed out.
Oh, we were signed out. Okay, well, people will have our contact information through the virtual booth. So anyway, thank you so very much. Dr. Davis, great job. Thank you so much.
You're welcome.
Bye. Have a great day. Good evening. Bye.